So hello everyone, my name is Mirjana Njegovan and I'll be your host for today. Welcome and thank you for joining this MDPI JCM webinar, the future of COVID-19 mRNA vaccines focused on innate immunity. If you wish to contribute to the topic of the webinar, please submit your research to the related special issue learning from COVID-19 vaccines. This is the link for the submission. I would like to remind everyone that this webinar is being recorded and the recording will be made available as soon as possible on the webinar's web page. Please note that you will have to be logged on into SciForum in order to view the recording. Webinar will have introduction by chair and free presentation of 30 minutes each. The presentation will be followed by a question time moderated by the chair. If you have any questions for our speakers, please post them in the Q&A box at the bottom of the screen. Please remember to mention the name of the person to whom you're addressing your questions. That would be here. Today's webinar is chaired by Professor Dr. Michael Goldman. Professor Michel Goldman is a highly cited clinical immunologist with a keen interest in vaccine safety. From 2009 to 2014, he was the first executive director of the European Innovative Medicines Initiative. Currently, he is the president of the Institute for Interdisciplinary Innovation in Healthcare at the University Libre de Bruxelles and serves as the editor-in-chief of Frontiers in Medicine. Michel Goldman is a laureate of the Lucien Steinberg Prize, shared with Peter Piot in 1997, and held the Spinoza Chair at the University of Amsterdam in 2001. In 2007, Michel Goldman was awarded the degree of Doctor Honoris Causa of the University of Lille. That would be all from me at this point. Thank you, Professor Goldman, for sharing today's webinar. And now I hand over the microphone to our chair. I hope you all enjoy.
Thank you very much and for this introduction, and hello everyone. Thank you for being with us this morning. Just as a short introduction, I think that we know actually since the pioneering experiments of Janeway and Medzitov in the 90s, we know that antigen alone is not sufficient to elicit efficient antigen-specific T and B lymphocyte responses. So what they call the little dirty secret, as you also know, has been clarified. And it has been clarified in the sense that we now know that activation of innate immune mechanism is critical to elicit efficient antigen-specific responses. In the case of vaccines, it is really the role of adjuvant contained in the vaccine to trigger innate immune cells in order to to elicit the response to the antigen contained in the vaccine. Now, before considering the innate immune responses triggered by mRNA vaccine, and that's the topic of today, we would like to focus on innate immunity triggered by COVID-19 mRNA vaccine. We think that it's very important to start by understanding the mode of action of, I would say, adjuvants, classical adjuvants in classical protein-based vaccine. We will start in a moment by doing so, and after that, we will move to mRNA vaccine, and we will discuss what is known and perhaps what is still unknown about innate immunity triggered by mRNA vaccine. Finally, innate immunity is important not only to elicit antigen-specific responses, as I just said, but also it is associated and linked to the phenomenon of trained immunity, which might be extremely important, especially for cross-protection against other microbial antigen, and this will be the topic of the last presentation. So let's start by discussing classical vaccine adjuvant. And for this, we are really very fortunate to have with us Nathalie Garçon. Nathalie has trained both as a pharmacologist and as an immunologist. And actually, she got 2 PhD in each discipline. She was trained at the Baylor Institute in Texas. And after that, and that's the reason why she's known by most of vaccinologists in the world, she has spent a number of years with GSK, starting with actually GlaxoSmithKline. And at GSK, she was especially in charge of, indeed, adjuvant technologies. After that, she joined a French biotechnology company, BioAster, and right now she's a consultant for different bodies. I will not make this introduction longer, Nathalie, and I will give you immediately the floor to discuss the development of classical adjuvants, I would say.
Thank you, Michel. Thank you very much. I'm going to share my screen. Okay. So good morning, everybody, and I look forward to this meeting. Can I? How do I get rid? Okay, I'm fine now. Okay. So as Michel said, I'm going to try to give a look in the retrovisor and look at what we have learned so far from adjuvant and what could be next. And I'll try to be concise and simple. There's one thing that we cannot forget is that an adjuvant alone doesn't mean anything. The adjuvant has to be with the antigen, and the antigen has to be relevant. And there has been the adjuvant is the conclusion, one of the conclusion of the 90s and 20s of a lot of innovation that appeared across time. And when vaccines were developed, there were empirical approaches, observation and repeating. The first really innovation breakthrough was cell culture and fermentation, and that was in the 93. The second one was molecular engineering and adjuvant, and that allowed the production of recombinant antigen, which do need adjuvants to be efficient. And there's a lot of vaccines today that are based on that principle. Then in the 90s, glycosylation started to be understood, and in particular, the ability to conjugate protein with sugar polysaccharide with a carrier, allowing to induce a response that was persistent and efficient. And in the 2010s, what's called reverse vaccinology was developed, and that was done by Rino Rappuoli, where the conjugation of bioinformatics, genomic transcriptomics, and allowed to identify in silico potential antigen and protein antigen in particular, and that allowed for the development of the main B vaccine. And molecular biology in the early 20s as a vaccine that was licensed and used allowed for the next step, which was viral vector vaccine, where the idea was to accelerate or simplify the production of vaccine. It's a virus that was expressing the antigen of interest in the body of the individual, allowing to simplify tremendously the production process. And one of the very big change, and that has transformed the vision of vaccine and has allowed the emergence of RSV vaccine in particular and COVID vaccine is crystallography and structural vaccinology that allowed to understand was after more than 50, 60 years of work on RSV vaccine, the vaccine could not work. And the ability to block the antigen into a form that could allow for neutralizing antibody completely transform the vaccine field and actually allowed for the vaccine, the RSV vaccine, to be developed and produced. And finally, RNA DNA vaccine, which is not new, it was in also in the 60s that the idea came, was really possible only now through the COVID pandemic vaccine, allowing to develop them fast and put the mean to an end. But the key to that formulation to that vaccine is a formulation, and that's somehow a common thread to quite a lot of the different technologies that allow for inducing and maintaining an immune response. It's the way you formulate the vaccine and the antigen that will make the difference. So if we look at the past and adjuvants, they were first discovered by chance. So there was a need, observation, and those were applied as adjuvant. The DNA was in 1885 with coli, the coli toxin that showed, even if anecdotal, effect on cancer vaccine. And then LPS, aluminum salt, all were designed and defined by serendipity and chance. And then in the 60s, the cooperation between T and B cells was understood. So there started to be a emergence and accelerated understanding of the immune response that allowed to have a more scientific approach to how to induce immune response without knowing at this time what was really the key. The emergence of HIV changed also the vision of adjuvant, and it was clear that alarm alone could not induce any protective immune response. So improvement on existing one like front adjuvant or the emergence of others became, and between 1986 and today, there are a lot of different adjuvants that have been licensed through different vaccines. SAF was the first virusome, and I'm not going to go through all of them, but they are all based on, few of them are based on one single adjuvant. This is the case of CPG for the hepatitis B vaccine. Most of them are combination with one or another. And the last one to be licensed actually is a combination of alum CPG for the COVID vaccine developed by Clover. So as Michel said, the dirty little secret was deciphered. It was clear that in order to induce an immune response that was an adaptive immune response, you needed different trigger, and that could only happen with the antigen and what is defined as the defense trigger or adjuvant that allow for the effective activation of antigen-presenting cells that continue in a cascade of events that lead to an effective immune response. And when you look at the different type of vaccines, the whole pathogen clearly is built in and does work very efficiently in inducing an immune response, but is sometimes fairly reactogenic. The next generation was non-replicating, so they are inactivated, killed, so that they do not replicate in a hope that it will be less, more the tolerability will be higher, which was true for some, not much for others. Then subunit, you take within the pathogen the part that you know is inducing a protective immune response, and that's what you use as a vaccine. For most of them, actually, they were carrying the natural adjuvant within their structure, so that allowed for an immune response that was still efficient, but not fully, and it was not sufficient for some pathogen. And at the end, in order, in an idea to ease as well the production of the antigen, protein recombinant, which is much easier to produce and in large quantity than virus were designed, and there it was obvious that adjuvant were required, and most of them, if not all of them, were used with aluminium. And then the emergence and the understanding of the dirty little secret and the fact that pathogen carried with themselves molecules that stimulate directly the antigen presenting cells, adjuvant re-emerge, and the question was, how can you do that with purified antigen, and getting back to an immunogenicity that was sufficient while keeping a tolerability that was acceptable. So if you look at what can be an ideal adjuvant, you do not want a bystander effect. And by that, you mean you do not want the adjuvant to induce an immune response against something that is unwanted. By design, an adjuvant is agnostic. He will induce an immune response against every epitope that the individual is capable of recognizing. So again, the design and the understanding of the antigen is key. It's nice that if you have the clear mechanism of action that allows not only to understand the cascade of events that will emerge when the adjuvant is given, but also that can help you to understand or prevent any side effect that can be interpreted or linked to a specific mechanism of action. What is clear also is effectively active humoral and cellular immunity with no adverse reaction across population would be a must. and it should be tailored to the population that best benefit from the vaccine. And you can clearly see that in the context of AS01, for example, the malaria vaccine is half the dose of the adjuvant that you have in the zoster vaccine, given its different pathogen and different antigen, but you adapt to the need. You do not blast as much adjuvant as you can. And needless to say, it has to be easy to produce in large quantity, easily stored and administered. So current adjuvant, they can be natural, so they come from microbes. TLR4 agonists come from microbe, or they can be part of plant. They can be hemisynthetic, so they're built on a natural molecule, or they can be fully synthetic. Clearly, and that was pointed out, and that will come again after me, the revolution in the understanding of how innate immune systems sense microbe brings a huge opportunity for their design and development. But it's not enough, because clearly that cannot, the understanding of pathogen-associated molecular pattern cannot explain all the effect of the adjuvants. So there are other things that are important. It is clear also that one size does not fit all. It has to be adapted to the antigen, to the target population, clearly, and the needed immune modulation for the vaccine to induce a effective protection. So what have they allowed to do? Well, they have allowed to overcome poor immunogenicity, and in the context of recombinant antigen, they have increased the duration of the immune response that has been shown in the context of HPV, with data of protection against cervical cancer that are more than 10-15 years today. And that has been, they have also allowed to induce what's called antigen sparing. So you can use less antigen with an adjuvant, allowing to do more doses of a vaccine, especially in the context of a pandemic. And they have improved the immune response in specific population. And the clear example are zoster and, as Michel said today, RSV, both with the same adjuvant, but at different doses. So understanding the present. There are many different types of adjuvants, and I will focus on the adjuvant that are currently in licensed vaccine. They can be only soluble. This is the case of CpG in the hepatitis B vaccine from Dynavax. They can be particulate. This is the case of aluminium with particles of a fairly small size, or they can be emulsion. Today, there's 2 licensed emulsion. They're basically the same principle with a difference in their composition that can give them higher effect in certain circumstances, or they can be combination. And those combination, actually, by the way, that's the case of AS03, where you have tocopherol, which is an immunomodulator as well, which is included in the emulsion versus MF59. So it is de facto a combination. And you have all the combination that have been first developed, the AS series, so the alum MPL, CpG alum, so that the clover vaccine, AS01, which is liposome with, again, a TLR4 agonist MPL, and QS21, which does not function through any known receptor. Matrix C and A, which is a cage-like structure with saponin, so the same type of molecule than QS21, which is in a COVID vaccine licensed by Novavax. So what do we do with the innate immune response that is induced by those adjuvants? In mice, it has been observed that all adjuvants, and not always tested in parallel, induce a potentiation of the transient inflammation response, that can be seen both at the injection site and the draining lymph node. And those effects lead to the stimulation of an adaptive immune response, a transient systemic response that with a landmark marker, which is CRP, transient systemic cytokine response, and a change in neutrophil monocyte and eosinophil blood fractions. Those studies in mice, and I won't go through them, I mean, they have all been published, have allowed to decipher the mechanism of action of those AS systems. And that really has been helpful in understanding better what we know and what we do not know in terms of induction of innate immune response. When you look at adjuvant in human, and to my knowledge, the only study that compared several adjuvants in the same study is the study that is described here, where aluminium alone, aluminium and MPL, which is AS04, an emulsion, and AS01 were compared in human with about 150 volunteers in each group, where the reactogenicity, the immunogenicity, the cell-mediated immune response, as well as the mediator, the induction of various markers in the blood were followed across first after the first dose and several hours or days after the first dose, and then post-dose too. And that has been very informative and somehow not surprising, but opening more doors and closing them. If you look at the antibody response below and the CD4 response above, and by the way, I don't think that I have not seen any adjuvant that is capable of inducing a CD8 response de novo with a recombinant antigen. That's a clear difference with mRNA. You can see that the protection in antibodies is considered to be 10 million international units. So clearly all of them do protect. There's no question about that. What you can see is that AS01, full or half dose, give a high antibody response. The emulsion gives a fairly high antibody response. The aluminium MPL does as well. And aluminium, which is a licensed vaccine, give a protection that is protective and long-lasting. So you do not need to use an adjuvant giving a response which is 1,000-fold higher than the current vaccine. It doesn't bring anything except probably reactogenicity. If you look, or you have to go in for specific population, that can have a sense. If you look at CD4 immune response, the difference are less, but clearly, again, it's the AS01 vaccine that gives the best CD4 response. And not surprisingly, this is AS01 also that is fairly reactogenic. Now, if you look at in the blood at a different time point from D0 to D37, and you look at markers of innate immune response, what has been seen is that CRP level were increasing, especially in AS03 and AS01 at day 1, and that's those here. So that's AS01 and AS03, and they were increased also for the other adjuvant only after the second dose. So here there was already a difference between those adjuvants. And when you look at the myeloid lineage, the neutrophil count increased after the second dose, and that was the most prominent signature that could be observed in that context. So if we look at the innate immune response more in terms of cytokine profile and blood markers, interestingly, all those adjuvants had the same key innate markers that were associated with the magnitude of the adaptive immune response. After the first dose, that was CRP, IL-6, and IP10, and therefore they were the ones that were most strongly associated with antibody response. And again, those were most prevalent within the AS01 formulation. Post those two, there were the strongest association of CD4 T cell antibody response was again with CRP and then IP10, and that was at the 31 and 33, and then interferon gamma and IL-6. So, and those were seen for every single adjuvant to different level, but they were the prominent marker that were detected. And despite the fact that there was really a question that was asked, there was no association with reactogenicity that can be observed or emerged from the statistical and meta-analyses that were done. So today it's still not clear why some people do have more reactogenicity than others, and an immunogen response that can be similar. So that study, which I find interesting, actually showed that different adjuvant can induce a common innate pathway. There's a canonical pathway that is associated with the enhancements of the adaptive immune response against recombinant antigen in human, and despite the fact that they are different in chemically and in terms of composition. So why is it so one can think that there is a base for the initial point of innate immune induction? Then there are probably many things we still don't understand, and that will be touched upon later, that clearly innate cells display epigenetic changes when they are activated, and that's what's called cranial immunity, and that can be differently activated and impacted by different adjuvants. So we have not yet deciphered completely what's happening. But one interesting point, and that's certainly something which is critical for LNP as well, is the formulation. If you look at AS01, the effect of the combined adjuvant molecule is superior to the sum of its part, which is actually the first time that, to my knowledge, that happened. If you take liposome alone, you have no response. And this is in specific T cells. You take MPL, you have something. QS21, you have less. And you combine them in the same formulation. That's when you have a synergistic effect with a clear difference in the response observed. What's interesting also, the effect of combined adjuvant can be different than their single part, and MPL works through TLR4, the TLR4 pathway. QS21 functions through the Cas1 pathway, and when you combine MPL and QS, you do see emerging a clear SYK activation that doesn't exist with neither of them, which is still not, to my knowledge, clearly understood, but certainly play a role in its effect. The persistence of the response is driven by specific molecule. I think there's no question today that MPL is key for the antibody response persistence, and that's what has been seen with formulation that contains MPL. Adjuvant can bring a pan protection and increase the breadth of the response. That has been seen with HPV, where two genotype in a vaccine can protect against more than 9, closely related. We see the same with the oil-in-water emulsion. So there is a capacity to enlarge the immune response generated by an adjuvant against a specific antigen, and that can be positive or negative. An adjuvant can protect better than the original pathogen in frail population, which was certainly, if not interesting, surprising in the context of zoster, where MPL QS21, so AS01, protect over 90% against zoster in people over 80 years of age. And that has been demonstrated up to 10 years so far in terms of protection. And today, the attenuated virus vaccine protect below 30% over the age of 80 years. So there is something there also that deserves to be understood. So the power of formulation, what next? Well, what do we have beyond current approach? Well, beyond the first line of defense, which are the pathogen associating molecular pattern, they are central to the induction of the innate immune response, but the interplay with different cell type involved in the innate sensing of adjuvant should be delineated and capitalized upon. It is not fully understood, and trend immunity probably is one of the answer. Optimum humoral and CD8 T cell response. Well, can adjuvant and mRNA approach combination be the best of both worlds when needed? Since adjuvant are very good at doing CD4 and antibodies, less so doing CD8. And beyond TLR, there are numerous other PR that have been evaluated as alternative immunopotentiator and potentially targeted alternative immune response. And STING for Th17 and mucosal immunity is a good example. And they are certainly opening the way to other arm of the immune response, alone or in combination, but also other paths of immunization, such as oral, but also intradermal as well. Persistence of the response. Can we build on the current knowledge of adjuvant use to educate on the key factor for immune persistence? Metabolic adjuvant, the new concept of metabolic state of myeloid cells impact on their innate response and capacity to stimulate T cells can certainly open a new avenue of immune stimulation through their optimum regulation and alternative candidate molecule. And the microbiome that somehow recapitulate all of that, and the recognition that the microbiome initiate and modulate the host defense function and can be modified in a way that its impact the overall innate immune response opens certainly the door to new approaches to adjuvantation, and this one is even further down in time. And CD8 T cell response, as I said, to my knowledge, there's no adjuvant that is capable of inducing de novo CD8 immune response in naive individuals. So can adjuvant or combination mimic live or mRNA COVID DNA vaccine in their ability to induce CD8 T cell response in human? I've not seen that. Current hypothesis is a combination of sustained presence of the antigen and optimal adjuvant signaling could lead to those effects. That has been seen somehow in animal model, not yet in human. So finally, to go back to the potential of combination, there's a paper that was published in 2016 where LNP adjuvant were tested in the context of dengue in non-human primate. And the interesting part is that they were compared to Iscomatrix, so it's a form of QS21 in cage-like structure, as well as an emulsion MA59. And there was a comparison of low dose of LNP. What's RCLs mean? It's a reduced cationic lipid, so you have the same amount of LNP but with less cationic lipid. And interestingly, what was seen is that in terms of titers, antibody titers, the Iscomatrix, the adjuvant, and the highest amount of LNP were the one that generated the most antibody. With here, this one is the LNP at the highest dose. This one is the Iscomatrix, and this one is the emulsion. However, when you look at the microneutralizing titer as well, that was a four genotype of dengue. Sorry, I should have said that. Oops. When you look at the cell-mediated immunity, it's a different picture. It is the LNP at the high dose that give detectable CD4 T-cell response, nothing in the others. And it is also the formulation at the highest dose that give the CD8 immune response. And it was established in that paper that clearly the cationic lipid play a critical role in that effect. So what is next? mRNA technology has clearly demonstrated its value for fast emerging responses. There's no question about that. The strengths, CD8 in naive individual, and the weaknesses with low quantity of persistence versus recombinant adjuvant and reactogenicity for the antibody have been highlighted, and one should remember that it has never ever been this is the first case ever that so many people have been vaccinated at the same time, and allowing to really show the strengths and the weaknesses of every single formulation, to a point where actually even when looking at the side effect, I don't know any database that had so much individual at the same time in the same environment, again the same circumstances, to look at the preponderance of some of those effects. And finally, it is possible today to combine existing adjuvant technology and new mRNA platform and to reach the best of both worlds. And that's an avenue that should be explored and that actually is being explored. Thank you.
Thank you very much, Nathalie. I think this was really an excellent introduction. You perfectly set the scene. And you keep the time, so that's really nice. So we can.
It was a challenge.
Yeah, I know. But I think that also at the end of your talk, you perfectly introduced the next presentation, which will be made by Rein Verbeeke, who is on the research group on nanomedicine at the University of Ghent in Belgium. Rein did his PhD there under the supervision of Stefan De Smet and Ine Lentaker. And actually, he has been since some time now being interested in mRNA vaccine and especially in the lipid nanoparticle, which are an essential component, which as we will hear, contribute to the adjuvanticity of mRNA vaccine. Actually, even during his thesis, he received several awards, and he made a very important postdoc stay in the laboratory of Peter Cullis at the University of British Columbia. And some of you may have read the excellent paper that they wrote in Immunity about what he will discuss today, which is indeed the activation of innate immune cells by nucleoside modified LNP encapsulated vaccine. So Rein, I will not do this introduction longer, and I give you immediately the floor for your presentation.
Thank you very much, first of all, for the introduction and for inviting me to this in my opinion, very interesting webinar. Also thank you, Natalie, for the great introduction. So I will immediately skip from the classical adjuvants to the mRNA vaccines and what we already know. And as I was already elucidated earlier, there are still a lot of unknowns. So we'll try to highlight this in my presentation. So first of all, a remarkable difference with, for instance, the recombinant protein vaccines is that the mRNA vaccines indeed do not require a further addition of an adjuvant or an immune stimulant. And we see in both in preclinical models as well in humans that they are quite capable of inducing both a T-cell response as well as an antibody response. Of course, this is a great feature of the mRNA vaccines, but it also raises a couple of pertinent questions, like, for instance, which components are then responsible for the innate immune response to the mRNA vaccines. As well, maybe there is, as it's maybe an uncontrolled adjuvant property that can be described with this vaccine. It's maybe also, there's also room for improvements. So basically, the question that I would like to address today is how do we think that mRNA vaccines are inducing an innate immune response, and then we can take the step towards the question how they should activate the innate immune response. I also need to highlight that the key findings that I will discuss are coming from in the review, as mentioned by Professor Goldman, that I wrote together with some great colleagues in the field. So for the people that are not satisfied after this meeting, I can refer to this review to go more in depth in the specific mechanism that we believe that can be ascribed to the mRNA vaccines. So first, take a few steps back. I will also try to go more in depth in the history, what we knew about the reaction of our innate system to mRNA. It was actually already in the early days after the discovery of mRNA that people figured out that upon infection of cells by RNA viruses, is that our innate immune response can respond to this and then in a way that it can inhibit the viral replication of the virus. It was also quite rapidly found out that it appears that this reaction, which was called the interferon response, seems to be playing a role in the metabolism of the mRNA, so the translation of mRNA, and that our innate immune response is capable of blocking viral translation in a way to defend itself. But then it took quite a while to figure out and what the specific immune mechanisms were for this interferon response. But then starting from the 90s with the discovery of pathogen recognition receptors, we now know that double-stranded RNA is a component which is recognized by, for instance, toll-like receptor 3. And we also have cytosolic receptors such as RIC-I and MDF5 and others. It was also figured out that in an uridine-dependent way that mRNA is recognized by toll-like receptors 7 and 8 in the endosomes, and that the cap structure on the mRNA is needed to avoid recognition by other receptors in the cell. The impact of this this RNA sensing, of course, if you think about mRNA vaccines and therapeutics, is quite an important one because, of course, the strength of inducing those kind of responses will be very important for your safety of your product. And as well as I elucidated earlier, the same like what you see with an RNA virus is that this interferon response can drastically lower the protein expression that you can obtain from these products. So if you don't have enough protein, of course, you will not have enough antigen to trigger your immune response. At least part of a solution came by the discovery by the people at the University of Pennsylvania by Kathleen Carrico and Professor Drew Weissman, where they figured out that if you compare mammalian mRNA to viral RNA or to the mRNA that you make in the lab, that a presence of post-modified nucleotides and specifically the uridines, that if the moment that you incorporate those kind of modified uridines that are present in mammalian mRNA inside your synthetic mRNA in your IVT mRNA, that you can drastically lower the recognition of your mRNA by the receptors that I elucidated earlier on this slide. And even so, is that for a long time that modified mRNA has been claimed to be non-immunogenic, and we still hear this. But then it's quite puzzling to see that those kind of modified mRNAs as well are used in our mRNA vaccines. So this brings me a bit to the beginning of the COVID pandemic. We know that in the vaccines that finally made it, so the Moderna vaccine and the Pfizer-BioNTech vaccine, is that they utilize the N1-methylpseudouridine modification, so the non-immunogenic mRNA, and this encodes for the spike protein encoded in a lipid-based nanoparticle system. But also at the beginning of the COVID pandemic, a lot of other kind of mRNA vaccine platforms already reached quite a substantial mature and other clinical trials and preclinical models. So we also have the unmodified mRNA, and I'm thinking about companies like CureVac, but also other companies are still focusing on the use of unmodified mRNA for vaccination purposes. And then we also have the self-amplifying mRNA. And what I would like to highlight in this slide is that if you look at the doses, I think this is the key finding is that you can administer much higher doses of the uridine modified mRNA compared to the other platforms, already indicating that the regenicity of those mRNA platforms is still a struggle for further development. Although for the attentive listeners or watcher of this webinar, there are also some indications that, for instance, an unmodified mRNA platform can induce similar responses compared to the modified mRNA platforms, which I will elucidate later on at the end of this presentation. But of course, it's important to highlight that mRNA vaccines are not only being developed for vaccination purposes. We also know that they are being used for cancer vaccines, where we want to induce another type of adaptive immune response, mainly focused on the CD8 T cell responses, but as well for protein replacement or for gene editing, for instance. Now, first products are going into clinical trials. Again, I would like to highlight, if you look at the doses that are administered, so I'm now showing here some of the pipeline of Moderna, is that you go to way higher doses compared to the mRNA vaccines if you look at those kind of products. Of course, also administration route can change, but for me at least during the pandemic and still reading the literature, it's quite sometimes puzzling to figure out how those seemingly similar mRNA products can induce very different kind of responses in our body, going from a strong cellular response to an antibody response, or to go into a product where you really want to avoid an innate immune recognition. So what I could go more into depth and let's say then the dirty secret about what's behind those kind of different mRNA LNP products. So first of all, a surprise came from a preclinical study in mice where the Pfizer vaccine was injected intramuscularly, where they found out that despite the use of the N1-methylpseudouridine, is that those vaccines are still capable of inducing a transient type 1 interferon response. And in this paper, it was also elucidated that this response, this type 1 response, was dependent on the recognition of the component that's still a little bit unknown, but it was figured out that MDPI-5, which is a cytosolic receptor for long double-stranded RNA, seems to be important for this response. So in mice, which have a knockout for this MDPI-5, you don't see this type 1 response. And then the next step, looking at adaptive immune responses, it was shown that it was not really important for the antibody response, but then looking at CD8 T cell responses, is that in these knockouts, either for the MDR5 or in mice models where you have a knockout for the interferon receptor, is that you can see a drastic drop in the CD8 T cell responses. We know that in the process of making mRNA, so using a bacterial T7 polymerase, is that this can give rise to contaminants. So it's not a perfect process. So there can be double-stranded RNA in your final IVT product. Of course, then it's important to use the appropriate purification methods. And as well, I think that depending on the structure or the mRNA sequence that you have, of course, you can also induce double-stranded structures in your mRNA product. What I would like to highlight here in this figure is that it was already known for a while that if you, for instance, inject, this is again in a mice model, if you inject a modified mRNA LNPs and systemically in mice, is that you can still see a quite robust pro-inflammatory response to those kind of processes. But the moment that you purify mRNA, so taking away this double-stranded RNA contaminants, is that you can almost completely mitigate this innate immune response to the products. And here, just to note, is that the mRNA was purified using a reverse phase chromatography methods. It was also recently shown by Moderna, where they indirectly maybe gave some insights in how they make the mRNA. But I guess it will also depend on the different kind of products that they are testing in their pipeline. Is that if you look at an unpurified mRNA, that you can still see double-stranded RNA the moment that you purify it here again with a reverse phase chromatography, is that you can lower the presence of double-stranded RNA, but it's not completely gone. And here they brought a solution where they basically start from the beginning. So how those kind of double-stranded mRNA contaminants are formed by mutating the T7 polymerase. And with their mutant T7, they can go to way lower levels of double-stranded RNA in their product. And if you then look again after injection in mice models, you can appreciate that the pro-inflammatory response here shown by the measurement of IP10 is that it can lower the response depending on the amount of double-stranded RNA that is present. Of course, this can make you wonder what the presence is of double-stranded RNA in the COVID-19 mRNA vaccines. I don't think that this has been tested or shown before. And then as the next step, it's of course important to know what the role is. If this double-stranded RNA plays a role in the reactogenicity or in the adjuvant effect of the mRNA vaccines. But of course, if you think about the use of mRNA products for other kind of application, I guess it's really important to get rid of this double-stranded RNA. The first insight that we can get for this is coming from a study that has been done in vaccinees of the mRNA vaccines, people that were known to have a deficiency in type 1 interferon response, and at least there it was found that the B-cell response was not reduced or was not significantly lower compared to the normal population. But this publication is silent about, for instance, the T-cell responses. Maybe there's a bigger role played for those kind of adaptive immune responses. So then brings me to the LNPs. So of course, it's not only the mRNA, we also have the LNPs that are an essential product of the mRNA vaccines, and there are also some surprises seen over here. So also going back here into history, so starting from the barrier, so the cell membrane, which is an essential part of our cell to avoid entrance of foreign materials. So starting from the barrier, we came to the carrier. So starting from simple liposomes composed of phosphocholine, people start to use this for gene delivery purposes, going to more complex systems. So first big deal was to have enough encapsulation of mRNA in a product. So people started to use cationic lipids that can interact with the negative charges that you have in your mRNA, giving it a bigger chance to encapsulate it in your carrier. Going to then more complex system, adding other kind of helper lipids such as PAC lipids, and then to the product that we know today, a 4-component system composed of an iron isolipid. I'll come back to that later on, phospholipids and PAC lipids. It's also important to highlight that those products were first designed for systemic delivery of small interfering or short interfering RNA, with the first product known as Onpattro, which came to the market. So there we know that upon administration in the bloodstream, that you have a sync effect with the proteins and lipids that are present in the blood. So that makes that your PEG lipid will come out. So the PEG lipid has also been designed with short tails that can easily diffuse out of the lipid membrane of your LNPs. And at that moment, you will have a lot of proteins that will adhere or that will have affinity towards your outer membrane of your LNPs. And it's known that this is an important step. So, for instance, apolipoprotein E is important to have the uptake by the hepatocytes, being recognized by the LDL receptor, and then going inside the endosomes. Is also important to highlight that after administration, your ionized lipid is in the physiological pH, so it will be neutral. It has been designed for this purpose, but at the moment that it comes inside the endosomes, you will have the acidification in the endosomal compartment, and this leads that the ionized lipid will become positively charged, and this is an important step to have a destabilization effect on your endosomal membrane, making that your gene product can come out and then reach its final goal to be translated, or here in this case, to have a knockdown of a specific protein. This is another example, but it was already nicely addressed by Nathalie in the first presentation. So this is indeed backed up by a few other papers, but this was then recently shown in the mice model again. If you combine empty LNPs together with a recombinant protein, in this case hemagglutinin for the flu vaccine, but if you compare this with other classical adjuvant that have been specifically designed for this purpose, that empty LNPs seems to have a quite strong adjuvant property. So going to higher antibody levels here compared to a classical adjuvant Adavax, which is a squalene-based emulsion. And this is a surprise because those products have not been designed for this purpose, and it was also then shown in the same paper. If you take out the ionized lipid by replacing it with a permanent cationic lipid, that you almost completely lose this property. In this paper, you also go a little bit more in depth, showing that empty LNPs are capable of inducing a quite robust IL-6 cytokine response. That this IL-6 cytokine response is important both for the induction of follicular T cells, as well for the induction of germinal center B cell responses. So indicating that at least the result of the innate immune response to the LNPs is important for the B cell response. Another paper also focused on more on the reactogenicity of mRNA vaccines, and what I'm showing here in this figure of this paper is that they used an ex vivo model, so using human PBMCs, which will mainly be consisted of monocytes. Is that the moment that you challenge the human PBMCs with mRNA LNP products, so comparing in this case, in this graph, an ionized lipids that is present in Onpattro products compared to S110-2, which is the lipid present in the Moderna vaccine, is that this can induce a quite strong IL-1 response. And that also even empty LNPs, you can see here the combination of the moment that mRNA is encapsulated compared to empty LNPs, is that at least the Moderna formulation seems to be capable of inducing the IL-1 response in this human PBMC model. I have to say that we have tried to mimic those kind of experiments in our lab. So what I'm showing here is a figure where we injected both mice as well as here in C, where we challenged human PBMCs with mRNA LNPs or empty LNPs. Again, in this case, using the Moderna lipids and our LNPs. So of note, if you inject those kind of material intramuscularly, you can clearly see a signal in the muscle, but as well here in the inguinal lymph node, you can see a clear signal. So this is after 6 hours of injection. And then comparing MT-LMPs and looking at cytokine responses, we see an IL-6 response, but quite low with MT-LMPs. And for other cytokine responses, it's only in the moment that you have the presence of the mRNA where you see a more clear pro-inflammatory response. So showing that at least the combination of both products, in my opinion, are doing the job. Also in the human PMCs, we are not capable of obtaining the same results as shown at the previous slide. So shown at MT-LMPs at quite relatively high doses, we're not inducing cytokine responses, but only in the moment that you have the presence of the mRNA is that you are seeing responses such as IL-6 and MCP1 and TNF-alpha and those kind of models. We also did a comparison by comparing an IS lipid, which is known to be more reactogenetic that was shown in other papers compared to Moderna formulation. So again, showing here the translation in the mice model, looking at cytokine responses measured after 6 hours of injection. Here you see that MT-LMPs are giving a more pro-inflammatory response compared to the Moderna lipid, but again, the moment that you have the combination with the mRNA, you see a strong response. Also looking at first safety insights, so looking at weight change, you can see that with the C12-200 lipid that you can see a drop in weight, which is not present in Moderna formulation. And looking at CD8 T cell responses, both compositions were capable of inducing CT responses with no significant differences, although there may be some trend towards slower responses with the Moderna formulation. More and more, we are seeing the same thing in new papers that are coming out, that people are trying to find less reactogenic ionized lipids. So it's still focused on the ionized lipids. I don't think that a lot of things are being changed now to the other lipids. But here is an example from a company, Genevant, situated in Vancouver, where they compared all the clinically relevant ionized lipids with their own property lipids, claiming that they can go to lower pro-inflammatory responses without losing the potent adjuvant activity to induce antibody responses. Also, what I mentioned briefly in the beginning, so showing that a modified mRNA was also capable of inducing similar responses in a phase 2 trial. So I showed it in the table in the beginning. Genevant is their LMP that has been used in those kind of clinical trials. So showing that both on the level of the mRNA as well as the LMP, that you can play with the innate immune properties. So to give an overall overview, so we're getting more or less a picture on what is going on. So after injection, we know that different types of cytokines are being produced, where it seems that the LMP can be a major contributor. We see that in the drainage to lymph nodes and what's happening in the lymph nodes, so having the antigen-presenting cells bringing information to T cells and B cells. Is that for the T cell response, at least, is that the type 1 response still seems to be important and seems to be there if you look at the mRNA COVID vaccines. And that then the IL-6 cytokine seems to be important for the induction of regular T cells and the B cell responses. I will not go into more detail because I guess this is more the expertise of the next talk. Is that we see some effects on monocytes, both in the bloodstream as well as other places in the body, where we see that there is an interferon signature in the monocytes, which seems to be there, and that we also see an increase in inflammatory monocytes and things like that. So I guess there are a lot of things that we still need to learn, looking more into depth and the different kind of cell types and then how they interact with the mRNAs and what they are doing to their program. So to conclude, so what's next? First, and this is maybe a personal frustration, but if you look into the literature, it's sometimes still unclear what people are using. So I guess it's important to standardize, and I really believe that the devil is in the details. So an LMP1 can be very different from an LMP2. If you don't disclose how your mRNA was modified. But I also elucidated the importance of double-stranded RNA content and purification, things like that. It will be very difficult to make conclusions, but at the same time, it's also an opportunity because this makes that we have some room for improvement, or that we can also, knowing the material design depending on the application, gives us room for tailored mRNA designs, as well that there might be multiple possible paths to develop an mRNA vaccine. So if you have better LNP, maybe you can use a unmodified mRNA or things like that, or if you want to make the self-amplifying RNAs working, maybe we need to put more efforts in LNP design and things like that. But there are also still a lot of unknowns. So the molecular mechanisms remain largely unknown. So the LNPs are doing something, but it's still unknown if this is in direct recognition by pathogen recognition receptors, if there are some things ongoing because of the fact that we are, in a way, disturbing the membranes to have the uptake of the mRNAs, things like that. So I guess there's still a lot of things to cover here. And of course, as let's say earlier, it's important to have to know the synergy between both components and what they are doing. And this will be important to of course note which kind of innate immune pathways that contribute to protective immunity. But of course, the next step is also to figure out is how those kind of innate immune mechanisms play a role in the reactogenicity and in the side effects that we're knowing. So also here, I'm looking forward to see more in the future. And with this, I would like to thank you for your attention and the people, of course, that helped me during the last years for this work.
Thank you very much, Rein. This was really a great talk, and thank you very much for also sharing some unpublished data, which I think are very informative. I think that now is the time to move to our last presentation by Mihai Netea. Mihai has been trained as a medical doctor in Romania and then moved to the Netherlands in Nijmegen to do a PhD. So he combined, clinical skills and also skills in, I would say, molecular biology. And he has been very interested in cytokines in general, and he did an important postdoc stay at the University of Colorado, and then he was back in Nijmegen, where he is currently the head of the Division of Experimental Medicine in the Department of Internal Medicine. I must say that importantly, he got in 2019 an advanced grant from the European Research Council, and is also a member of the Netherlands Royal Academy of Sciences. So Mihai is really one of the best experts of the so-called trained immunity. So without further ado, Mihai, you have the floor.
Thank you very much for the kind introduction, and also thank you very much for inviting me to talk today and present some of our data. So I will immediately share my screen. So I hope that this is to be seen easily by everyone. So I'm kind of the exception that's, let's say, the odd person today in the between the speakers, because my primary knowledge and expertise was not on the vaccines, and I more or less stumbled upon the vaccination field approximately 10 years ago, and I'm coming from a very different angle, that angle being innate immunity. I'm an infectious diseases specialist, and in my main expertise was always trying to understand modulation of innate immune responses in severe infections, fungal infections, and sepsis. But at the same time, I was working together with one of my colleagues regarding innate immune responses to mycobacteria. And through our work, we came into contact with some very old literature regarding the BCG vaccine, Bacille Calmette-Guerin, which was developed 100 years ago, actually. There was an anniversary short ago, and it has been one of the most successful vaccines Of course, one of the oldest vaccines at the same time. It's an attenuated microorganism. It gives a nasty scar, let's say, after being given intradermally. But still, it's one of the most used vaccines in the world because it was very useful for the protection against especially severe form of tuberculosis in children, TB meningitis, disseminated miliary TB, less effective for pulmonary TB in the adults, and so on. Nevertheless, BCG, again, one of the most used vaccines in the world, approximately 120, 130 million children receive it every year. But something very interesting, we came in upon some old literature, something very interesting was observed, namely that BCG, when it was started to be introduced in various countries, it induced a very strong decrease in overall mortality in the young children when it was administered. This is one of the first reports coming from Sweden in the 1930s, so almost 100 years ago, reporting that mortality in the children decreased from 11 to 4% after introduction of BCG. And this could not be explained actually by the TB deaths, because those were responsible for less than 1% of mortality in these children. But it was more like neonatal sepsis, non-tuberculous respiratory tract infections that were significantly decreased. And this observation was made many times when BCG was introduced in various countries. And it was also reproduced in clinical randomized trials in the end, done in the last 20 years or so. So we started to become interested in that, because we thought, well, as the people speaking before me have very nicely shown, T cells and B cells are very important, and those are the cells that we believe are crucial for the protection given by vaccines, but they are antigen dependent. So how can we protect against overall mortality, against other types of respiratory tract infections with BCG? So first of all, we were interested to see, can we reproduce some of these data by doing an experiment, an experimental study, experimental clinical trials in humans, and showing that BCG vaccination can protect against another type of infection. And of course, it's not easy to infect human beings, but we have used actually yellow fever vaccine as a model of very mild infection, because yellow fever vaccine is also an attenuated virus. We can measure with a PCR the number of viral particles in the circulation. So what we did, we asked two groups of volunteers to receive either BCG or placebo, and then 1 month later, everybody received yellow fever vaccine. And we just assessed by PCR the viremia in the blood of these volunteers. And as you can observe here, the yellow fever viremia on day 3 and day 7 was low in very few individuals, because it was too early in the beginning, and thereafter it was eliminated. But most of the people had viruses in their circulation on day 5. And you can see here in this logarithmic scale that the viremia actually in the BCG vaccinated individuals was significantly lower compared with the placebo individuals. And that was not associated with any defects in antibody titers later on day 90. The antibody titers were perfectly fine in the placebo and BCG vaccinated individuals, meaning despite the lower viremia, antigen presentation with the viruses that were available was very good, very nice. Antibody production was normal, so everything was fine. So that did not lead the previous BCG vaccination to a reduction in the specific immune responses, but at the same time it did reduce the viremia. So this demonstrated that BCG can reduce, actually can induce at least a partial protection against, of course, a very mild viral infection now, but nevertheless a very heterologous type of protection, because antigenically speaking, the yellow fever virus and BCG have very little, if anything, in common. So we were interested, how is that possible? If it's antigen agnostic, let's say, this response, would it be possible that we have also long-term changes in innate immune responses, which are, of course, antigen non-specific? And we stimulated monocytes from these individuals with Mycobacterium tuberculosis on the one hand, and 2 weeks later, this is another study, by the way, and we collected blood before BCG 2 weeks later and 3 months later, and we stimulated with either specific antigens like M tuberculosis specific pathogens, or also non-heterologous pathogens, Staph aureus and Candida albicans, again, having nothing to do with BCG. But we have observed that the monocyte production of pro-inflammatory cytokines was increased after BCG vaccination, also after heterologous stimulation. And we repeated that several times, and I'm not showing all the data because of the time, and I want to go to some new data. But the conclusion was, indeed, there is a change in the function of innate immune cells, and we already know that there is some function change in the capacity of the cells to present antigens, for example, after certain vaccines and so on. Beautiful talks about adjuvanticity and so on before me. But what we clearly were able to show that also the direct antimicrobial function of the innate immune cells was also changed, was also improved, and that was antigen agnostic. And how is this working? What we have observed is that there are metabolic and especially epigenetic changes which are very important in these myeloid cells after the vaccination, but also after certain infections. So what do I mean by that? So DNA is very tightly packaged in the nucleus of a naive innate immune cell, such as a myeloid cells, a monocyte, or a macrophage, because we don't need to, in these regions which are important for host defense, we don't need to produce cytokines and chemokines and defensins and so on. So you keep the DNA closely packaged. It's like keeping the book with all the information closed, because you can walk around much easier with a closed book. Now, if you encounter an infection, if the cell is activated by a certain pathogen, well, gene transcription is needed. Cells start to produce cytokines, defensins, and so on. This is allowed actually by chemical modifications in the histones, methylation, phosphorylation, acetylation, and so on, which induce a change in the architecture of the histones, which are now much more much less compacted in the nucleus, and that allows the transcription factors to bind to the promoters of the genes and induce gene transcription. Now, after the infection is eliminated, we've always thought that this loosely form of chromatin architecture is changed, is going back to a closed conformation, because all these chemical changes in the histones are lost. And this is partially true. We do go partially into a close conformation architecture of the chromatin, and a lot of these changes are lost indeed. But especially the methylation of these histones do persist, and they are like a bookmark in the places that were initially activated. It's like closing the book because you don't need it to be open, but you put a bookmark at the right page. So when you encounter again a pathogen, you know where to open the book. And you know how to induce gene transcription much easier on the right place and much more efficiently. And this leads actually that the monocytes and myeloid cells, after stimulation with certain microorganisms, after certain vaccines, they are able to respond better later on. And this is due to this epigenetic reprogramming of the cells. Now, there are very complex, let's say, interactions and changes that take place in the cell that are responsible by these that are responsible for this functional reprogramming of the cell. And there is an interaction between pattern recognition receptors inducing intracellular signaling pathways, in which we have shown that PI3 ACT mTOR pathway is very important. That leads also to changes in the metabolism of the cells. There is an increase in glycolysis to produce more ATP. At the same time, there are changes in the TCA cycle. I'm showing only conclusion of several years of studies. The citrate is extracted from the TCA cycle. This forms acetyl-CoA, which is important as a donor for acetyl group, for histone acetyl transferases, for these epigenetic changes. At the same time, there is cholesterol synthesis pathway stimulation, and one of the important metabolites here is mevalonate, which provides amplification loops through IGF-1 receptor. And at the same time, for example, there is accumulation of fumarate through glutaminolysis that inhibits histone demethylases, and that allows that the histones remain methylated for longer period. And there are also important roles for low coding RNA, which serves as transporter for the methyltransferase in these processes and so on. But there is this interaction between immunological signaling, metabolic changes in the cells, and epigenetic reprogramming that allows the cells to maintain a primed, trained, we called it, state, which allows them to react better later on. Now, this is very nice. We did this after BCG vaccination, and later on also we did after influenza and after MMR vaccination, which were also shown to have some heterologous effects. But how can we explain that monocytes in the circulation are changed, and after 2 weeks or 3 months and so on, we went even up to 1 year. After 1 year, it starts to wane. This is a very important message. These changes are not as long-lived as the changes that take place in the B and T cells. They are shorter, but still for several months, up to 1 year. But how can we explain that? Because the monocytes are in the circulation only for a couple of days. So the only possible explanation is if similar changes are taking place in the progenitors of these cells in the bone marrow. And for that, we did a proof of principle study in which we asked people vaccinated with BCG to donate blood, not only blood, but also bone marrow aspirate before BCG vaccination and 3 months later. And we purify the hematopoietic stem cells from these BCG vaccinated individuals, and we performed RNA sequencing. We performed also ATAC sequencing to look at the chromatin accessibility. And what we have observed basically is that the hematopoietic stem cells had a very clearly changed transcriptional program after BCG vaccination, 3 months after BCG vaccination. At that time, BCG is eliminated. We were not able to find it at the site of the vaccination, not able to find it in the bone marrow or in the blood, but the transcriptional program is changed. And that is changed in the direction of myelopoiesis and granulopoiesis very clearly. And that is probably very likely responsible for the changes that we see later on in the circulation, because this is where monocytes are coming from. Now, in the next step, we ask ourselves, well, we change this function of the monocytes. Okay, but do all the monocytes change in the same way? Or do we have subpopulations of monocytes which are changed differently? And for that, we purify the monocytes, we expose them to BCG for a short period of time, for 1 day, and then we let them rest for 6 days in vitro, and then we re-stimulated them with LPS. They respond stronger after being trained or primed with BCG. But then we perform single-cell RNA sequencing to see whether there are subpopulations within these cells. And what we observed, yes, indeed, there are subpopulations. There are some of the monocytes which respond in the same way after heterologous stimulations, even if they saw or they didn't see the BCG in the first place. So basically, they respond to stimulation, but in the same way. We call them non-trainable. They were not really trained. Then we saw some of the monocytes that upon re-stimulation with LPS, they produce much more chemokines than the cells which did not see BCG previously. We call them monocytes producing chemokines. And then there were some half of the monocytes that produce both chemokines and especially pro-inflammatory cytokines such as IL-1, IL-6, TNF, and we call them monocytes producing chemokines and inflammatory cytokines. Now, by being able to identify these monocyte subpopulations, we also were able to identify the transcriptional programs which underlie these changes, and we were able there computationally to look in the databases, in the literature, the databases of single-cell sequencing, to see are there diseases in which these programs are either overrepresented or underrepresented. We looked, for example, in inflammatory bowel diseases, in Crohn's, and we have seen clear upregulation of these programs. We are not going in that direction, but what I want to say is that inappropriate activation of these programs can lead also to hyperinflammation, deleterious effects, and so on. But then we looked at COVID-19, and this were not our data. These were two independent cohorts collected by colleagues in Germany, in which they did single-cell sequencing in the circulation to observe the changes that take place in COVID-19 cells, and we extracted the data for monocytes from these two cohorts, and we analyzed in individuals with mild and severe COVID-19 whether they are able to express these trained immunity transcriptional programs. And what we have observed is that yes, the individuals with mild COVID-19 were perfectly fine in expressing these programs. They were even increasing it, especially the MCI program. But individuals with a severe COVID-19 were not able to express properly these transcriptional programs. So we hypothesized based on that there are two situations actually which are encountered during an infection. If somebody gets an infection, in our case it's COVID-19, but it can be also other infection, and there is a high proper effective innate immune response in the beginning when the host encounters the pathogens, the pathogen multiplication is inhibited. That results in low pathogens load, in our case low viremia, for example, low systemic inflammation, low systems, and survival of the individual. However, if somebody gets the microorganism when they are home and they don't know even that they are infected, but there is a poor innate immune response in the beginning, there is a quick multiplication of the pathogen, high pathogen load, high viremia, high systemic inflammation, which now it's ineffective. That leads to high symptoms, high severity of the disease, and unfortunately, some individuals could even die. So with this in mind, we were thinking, would it be possible to use these? And we were at this moment at the beginning of the pandemic in 2020, would it be possible to use this knowledge to try and even partially protect, for example, against systemic viral infections also in the adults, like we have seen the epidemiological studies in children. And we did a study, the Activate study, which has been done actually just before the pandemic in 2019. Before we knew that, there was a study done in elderly individuals who were hospitalized for other problems. They were going home, and we know that people after hospitalization, they are in an immunosuppressive state. They have a high susceptibility to various infections, and we randomize them to receive either placebo or BCG, and we follow them for 1 year. And we got the results beginning of 2020. And what we have observed is that indeed the BCG vaccinated individuals had approximately 40% less infections diagnosed compared with the placebo vaccinated individuals. So that was promising, but this was before the pandemic. So this could be due especially to influenza or other respiratory viruses and so on. And then we hypothesized together with other colleagues in the field that maybe BCG might be able to protect maybe against SARS-CoV-2 as well. And at that time, beginning of 2020, we didn't have yet the specific vaccines, which were expected to protect much better, obviously, because they are specific. Some people did mouse studies. This was a study done by Alan Scher, and he was able indeed to protect against BCG, you can see here. But there was kind of a problem that we were seeing already at that time, is that it was BCG intravenous in the mice that were able to protect against the lethal SARS-CoV-2 challenge, but not the subcutaneous. And we know that in humans, we give the BCG intradermally, not subcutaneous, but still not intravenously, obviously. So we started a number of studies actually to investigate the effects of BCG. We did a first study of BCG revaccination in elderly in Greece, and I'm saying revaccination because the population in Greece got the BCG at birth. And following them up in the beginning of the pandemic for 6 months, we have seen a decrease in the number of diagnoses in the BCG group. And with this promising data, we thought, okay, now we really have to do a proper big study. And we performed the BCG prime study in the elderly in the Netherlands. In the Netherlands, there is no vaccination at birth. So this was the first time that the people got the BCG. And we followed them up for 6 months again, and now we follow them up also for 1 year. But unfortunately, we were not able to see absolutely no effect of BCG in terms of the total number of COVID-19 infection. So, and these similar data were reproduced at the same time in South Africa and Denmark. So apparently the BCG was not able to produce, to protect against the total number of infection. However, what about the immune response during the infection if people got COVID-19? And interestingly, what we were able to see is that the BCG vaccinated individuals had better cellular responses, but also better serological responses. You can see here in the 3 panels here on the line C, it's the anti-SARS-CoV-2 serological response, and that was seems to be better. And we thought, well, maybe we cannot protect against the total number of infection, but maybe we have slightly better a better outcome in terms of severity of the infection. Yes, before going there, in parallel, there were colleagues actually looking at BCG and the impact on the serological and cellular response upon the SARS-CoV-2 vaccines that were starting to be developed at that time. This is a study with the Pfizer vaccine, and people who got BCG 1 month before the SARS-CoV-2 vaccine, the Pfizer messenger RNA vaccine, they were observing better neutralization titers against the virus. And this is also a BCG revaccination study done in India by colleagues from India and UK with the adenovirus SARS-CoV-2 vaccines from AstraZeneca, and they observed the same thing. People, if they got 1 month before BCG, they got both better serological responses and cellular responses. So this seems to be a pattern that was repeated, that if you had BCG, and we know BCG has adjuvant effects basically, but people responded better to SARS-CoV-2 vaccines as well. So then we looked at the severity of our individuals in the BCG prime study. Now we didn't have enough hospitalizations and deaths, fortunately, in our study, but still we were observing that there were 15% less hospitalizations in the BCG group. Very interestingly also, and I'm not going now into much detail, but it seems that also in this study, BCG was much better able to protect against influenza than against COVID-19. And that was beautifully reproduced by a study published in experimental models by Maziar Divangahi from McGill University, showing that the BCG in experimental studies protects very well against influenza, but very poorly, if anything, against COVID-19. So this is a very interesting difference between the pathophysiology of infection, so which also shows that these heterologous effects are not completely non-specific. You cannot protect with BCG, for example, against everything. It depends a lot on the pathophysiology of the secondary infection. We looked also at mortality, and I have to say very clearly, this was not power for mortality, was not meant to look at mortality, but we just looked to see what are the numbers there. We have seen in our BCG prime study that in the placebo group there were 18 deaths, in the BCG group 13, and then we asked and put together all the data from other BCG trials, and what we observed actually, and again this is summary statistics that was not meant for this type of analysis, so we have to take this with care and not to overinterpret the data, but it seems that in all the available studies, there is an advantage in terms of mortality in the BCG group, and per total there was approximately 40% less mortality in the BCG groups compared with the others. It's not only BCG which has this heterologous immunological and clinical effects. Beautiful work done by Bali Pulendran's group has shown that adjuvanted influenza has very interesting and important effects on the innate immune cells. It induces a program of antiviral vigilance, which again contains mechanisms at epigenetic level, leads to increase in antiviral genes and increased resistance to infection. And in some epidemiological studies, but unfortunately there are no clinical randomized trials, it seems that during this is a study that we have done in our hospital, individuals who were vaccinated for influenza had approximately 40 to 50% less infections. But of course, there is also a healthy vaccinee bias here, so we should take this data very carefully. Very interestingly, and now we go to the new discovery of the new vaccines, and we have heard beautiful talks about the new SARS-CoV-2 vaccines. People have started to look also whether the new vaccines might be able also to influence innate immune responses. And Bali Pulendran's group, again, they have done several studies. This is a study in Nature in humans. They were publishing also a Nature Immunology paper in mice, and they have observed also very important changes at the transcriptional and functional level, not only in adaptive cells, very important, of course, but also in innate immune cells. Long-term changes induced by the messenger RNA vaccines in innate immune cells, strong increase in interferon pathways, but also other innate and inflammatory pathways, as you can see in this graph, which is extracted from their Nature paper. There are also colleagues in Ireland who have studied in depth the trained immunity, so the heterologous effects on innate immune cells induced by the adenoviral vector COVID-19 vaccines from AstraZeneca, and they again observed not only antigen presentation, which is increased, but also an increase in the function of the monocytes, metabolic changes, and functional changes in terms of production of chemokines and cytokines by the cells isolated post-vaccination. So it seems that these new vaccines that are very important, they induce very important effects on the adaptive level, but they also lead, as many other vaccines, also to important changes in innate immune cells. And I think this is very important to understand and use in the future. Because how can this be used? We now see basically that various vaccines induce what we call trained immunity, changes in the function of innate immune cells. They might also induce, through their adjuvanticity effects, for example, improvement in the responses to anti-COVID vaccine. They also induce actually improvement in heterologous T-cell responses. And it seems that this is also an innate mediated mechanisms through cytokines such as IL-12, IL-15, and so on, that lead also to improvement in some heterologous T-cell responses. And how can we use that? So first of all, one could argue, for example, that in a future pandemic, when there is a new pathogen for which we do not have yet specific vaccines, until we develop them, test them, distribute them, and so on, we can try to use to perform very quickly phase 3 clinical trial with some of these vaccines that induce heterologous effects, to see whether there is even a partial protection. 30–40% probably is the most that we can hope, but still that can alleviate some of the suffering of the people until we come with specific vaccines for the entire population. And of course, specific vaccines are always expected to be much better, and they will be the preferred choice. But until we have them, we can use very quickly some of these heterologous effects. One possibility, of course, and I think this is something that we should aim in the future, is to try to build future vaccines that could incorporate both the adaptive immune memory, which is very important, for which they are built, but also some components of this innate immune memory that could give some heterologous protection. And somebody can think of combining antigens from important pathogens with adjuvant, but also with inducers of trained immunity to obtain vaccines that can be more broadly effective. So what I wanted to share with you today is the fact that vaccines, also infections, but I didn't have time to go into that, but several vaccines are able to induce epigenetic and functional reprogramming of innate immune cells, and this is likely to have interesting and important effects against heterologous infections, and that hopefully can be used in the future to build improved vaccines. With that, I would like to thank, of course, all the colleagues from our group and from the various groups with whom we collaborate, and thank you very much. And if anyone is interested in this subject, there will be at the end of this month in Naples a conference on that. Thank you very much.
Thank you so much, Mihai. I think that this is indeed was a nice review and discussion about trained immunity and its relevance not only to COVID vaccines and COVID, but to infectious diseases in general. So thank you very much for this. I will check in the Q&A.
I see that there is a question for me, if I can already say that there is a colleague, Florencia La Greca, who asks whether there are also effects of the people who were earlier vaccinated with BCG, for example, at birth or much earlier, not 1 month before, and whether that influences also the responses against COVID. In our data, it doesn't. There are some ecological studies suggesting that in countries with BCG vaccination, the COVID is less or whatever. I think that those studies are very likely to have so many biases that it's very tough to be able to conclude that. In our study, we do not see any protection or improved responses if people had BCG vaccination at birth. I do not think that this type of protection can go beyond 1.5 years.
Thank you. I have, in fact, a related question, because your data were collected in the elderly, and we know that in the elderly with aging, the process of methyltransferase might be affected in some individual by this phenomenon, which is known as clonal hematopoiesis, which involve, for example, the NMT3 and TET2. So I'm wondering whether part of the heterogeneity that you see in your studies might not be related to the fact that some of the people that you investigate actually present with clonal hematopoiesis.
This is an excellent point. We have not been able to look directly at the relation with clonal hematopoiesis. What we have seen is actually that the responses are somewhat more muted in the adults compared with the children. We see also, if we look epidemiologically, I think that is also a strong suggestion that BCG, even at heterologous level, works better in young children than in the adults and the elderly. We know that in the adults, we have seen that only half of them respond efficiently to BCG with an improved responsiveness thereafter. We have studied now recently a cohort of 300 individuals in which we were able to look a little bit of the efficiency of the response and inter-individual responses. It's only half of the people who respond properly to it. So BCG is useful, but it's not a very good vaccine in that respect. It has been very useful, but I think there is a lot of scope actually for improvement. And in terms of the relation to clonal hematopoiesis, this is a very good question, but I don't think it has been asked yet, but very, very interesting.
Thank you. We have another question from Frank Kulstart, who is from the Belgium Clinical Center. And thank you for being with us, by the way, Frank. You asked a question, do some adjuvants have similar effects as BCG? Perhaps this is a question for Nathalie.
Thank you. Actually, there's a paper that was published, I think, two or 3 years ago, looking at the AS01 with hepatitis B and looking more in depth into the profile that were generated both in terms of, and I was especially looking at different BTM. The difficulty here is that those BTM are specific to specific clustering of gene and effect, and they've been defined with cancer, not for vaccines. So that does give some bias in the clustering that is being done. But nonetheless, there has been, they looked at metabolite clustering and things like that. And you do see, and this is for AS01 in particular that is being seen, but interestingly, some of the BTM are shared with AS03. And those do not share anything, they have nothing in common. One is an emulsion, the other one is a liposome with 2 immunomodulators. So there has been some that has been seen, but certainly not to the extent of what MIAI has done for BCG. This is not really surprising. You, at least for AS01, you do have constituent that the LPS derived is one of them. So, but that has not been seen to that extent, and not both in terms of spread of what has been seen and intensity. But some effect on trained immunity has been seen with the AS01 in particular.
Thank you, Nathalie. I see that we have another question from Patrick Couvreur, who is working on all these questions at the Université Paris-Saclay. He's really a leader in this field. So his question is for Rein Verbeek. The mRNA and LP are inducing an excellent protection, but don't avoid contamination due to absence of IgA production at the mucosal level. Could you comment on which way nanoparticles could be designed to allow also mucosal immunity?
Yeah, thank you for the question. First of all, it needs how they are designed now. They are not capable of if you change the administration route to mucosal routes, the mRNA vaccines will not work. We also tried to test that, and you're not capable of delivering enough mRNA if you go intranasally or intratracheally with the current composition of the nanoparticles that are used in the mRNA COVID vaccines. Although, of course, it could be of interest. We see that the mRNA COVID vaccines now are to low level capable of inducing mucosal responses and IgA responses, but of course there's room for improvement there. I think the biggest hurdle is the mucosal layer. So it's a difficult layer to cross. There might be people are using now mRNA LNPs for lung delivery, but it's still quite difficult. And give like one solution. I don't know what I could propose, but it's yeah, there's room for improvement to design LNPs that are capable of doing this job, but at this moment they are not capable of doing that.
Thank you, Ray. Now we have a question from Alain Lemoine, who is the head of the Department of Nephrology Transplantation at the Hôpital Erasme in Brussels. So his question is an increased risk for autoimmunity or alloreactivity really in relation with trained immunity. So that's for you, Mihai, I guess.
Unfortunately, it is. So we have seen, for example, that if you activate these processes inappropriately and at the wrong time in the wrong circumstances, you can get inflammatory reaction that can lead to deleterious effects. That has been shown for atherosclerosis, for gout, for lupus, and so on. There is also beautiful work done by the group of Jordi Ojando at Mount Sinai in New York, showing that these processes actually can influence even also organ transplantation. And of course, we have studied ourselves innate immune cells because we are interested in that. But during the induction of these processes, there is an entire interaction between innate immune cells and T cells, for example, very important. We know that these processes lead also, as I showed, to improved T cell and serological responses, and that is beneficial. But at the same time, in other circumstances, if we inappropriately activate these processes, that can lead to inflammatory reaction. And even it has been shown to be very important for rejections in transplantation and so on. So people are now trying to inhibit both T cells and inflammatory responses in rejection and so on. So yes, unfortunately, we pay always. There is no free lunch in life, so we pay with the possibility of deleterious effects if we activate these mechanisms inappropriately.
No, actually, this mechanism might probably explain some types of graft rejection that we see in the course of infections such as CMV infections. So,
yes, what is very important, for example, I do think for the new vaccines that have been developed is that train immunity is not one single program. There are very different programs. So what BCG is inducing is not what influenza vaccine is inducing, and not what the new vaccines are inducing. So I think it would be very important also for the new vaccines to characterize that and to understand, is it advantages or is it disadvantages? I have seen one very interesting study from Hong Kong in which they looked, if I remember correctly, at the messenger RNA vaccinated and unvaccinated people, and they have seen less TB reactivation actually in the COVID-19 vaccinated individuals, suggesting that these long-term effects induced by the messenger RNA vaccines might be protective for other things that we don't think now. And it was a very interesting epidemiological study. So on the other hand, you can also think in certain individuals with a certain genetic makeup or so on, it might be responsible for a couple of the rare side effects. So I think it's an area that we really need to study to try to understand. And now is the chance, because now we have the tools, and there is at least two very important platforms which have been developed: messenger RNA, adenovirus vaccines. And I think it's very important to try to understand also these effects on innate immune responses, in addition of course with trying to get the best possible adaptive immune response.
thank you very much. No, I have a question for the three of you. You show several, very interesting and important data. Some of them were collected in mice, other in humans. And I'm wondering whether we could always translate what we see in mice to humans. And I would like to start with Natalie, because I think that your comment on CD8 T cell response was appropriate regarding this.
How would I say that? There was a study that it was not published, but going back to all the data that were generated in animal model with the different adjuvants, and doing a look at those data versus what was seen in human with those different adjuvants. Actually, the classification of the adjuvants in terms of type of induced response with looking at and I talk about recombinant protein antigen. The type of response that was induced across the board with the adjuvant was pretty much the same with different antigens. So, and the classification was always the same. The ranking of who was better for what was always the same in mice. And actually, it was the same in human. The difference was the log, the differences in terms of intensity. So you could have a tenfold difference with one formulation versus the other. You had less differences in human when you were looking at the data was generated, but the classification was always more or less the same. That's being said, that was at the time where system vaccinology and translational science was just words and not really looked at. So maybe that would be different. But I think in general, despite the common dogma, mice do not lie all the time. Rabbit do. I mean, we see that for reactogenicity, but mice, they do not lie that much, and it is especially true, or that can be not true when you're looking at molecules that act on specific receptors and they're not expressed the same way or on the same cells between mice and human. CpG is a good example. CpG is expressed on B memory cells in human, it's not in mice.
Yeah.
And TLR receptor expressed in B memory in mice, it's not in human. So here you can see differences, but when the expression of the receptor is the same, it's there's no really difference. And when the mode of action, the way it works like QS21, it's through interaction with membrane cholesterol. This is agnostic, it doesn't change between species. So in that term, that not much difference has been seen. There's more diversity in human, frankly. I mean, individuals do make a difference in the response.
Yeah,
So,
if I
perhaps a
may
question
just
for Rain, I think that there was some differences in the response to MT-LNP in mice and humans. So could you elaborate on that?
Yeah. Maybe first, in general, I guess I agree with Natalie. I think that looking at what we learned for the mRNA vaccines in preclinical models and in mice, I think it has been quite instrumental to what we have learned after the COVID-19 pandemic. Like, in my opinion, you can definitely use the preclinical models to make predictions or to compare different kind of products or different kind of vaccines. And that way, I also don't think that they lie. Your question for the MT-LNP, so the data that I showed in the presentation, so then we used an ex vivo model, so we used human PBMCs. And also in our hands, I have to say that the correlation was really there. So if we compare again different kind of materials, we see the same things going on in mice. Although what I think that is maybe a big difference is that it's maybe more related to dosing, is maybe the same mechanisms are there. But what I also learned from colleagues, like people that are using monkeys, is that monkeys, they don't, the mRNA vaccines, they don't give any harm. So they can handle it. It's all fine. They don't see side effects in the monkeys. But if I look at humans, at same doses, it becomes another story. Or in mice, the doses that we are applying are much higher than compared to what we are applying and the other models are in the humans. So there maybe is more bigger difference over there. But I guess that the innate immune mechanisms, depending on the species, I think there are a lot of things to learn in the preclinical models that we can take and that can make predictions for using the same materials in humans.
Thank you very much.
Michel,
Do you want to come
actually,
here, Natalie?
I had a question for actually, and I was on that line because do we know exactly? So we know how much mRNA we inject. We do not know really what is the cargo load in the structure. So how much of what we inject is really effective as a.
I think that it's the doses for the mRNA vaccines have been. It's in the final product that it says. So it's the mRNA that is encapsulated in the mRNA vaccines.
Yeah.
I guess this is the doses that are shown in the publications or and are probably also the doses that are used in humans. So it's in the final product that they measure the encapsulated mRNA that is there.
Okay. And there's none outside of the LNP or. Okay.
Of course, it will not be. You don't have a perfect encapsulation, but I think that it's at least that is how we how we show it in the publications is that the doses that we find encapsulate in LNPs. So that's how we do it. And I guess it's the same for others, but I'm not sure.
Thank you very much. So there is no more questions from the audience. So yeah.
You wanted may I opinion on your question?
No, I just wanted to add that we are sometimes making, in terms of the mouse models, we are making our life a little bit difficult also because we keep the mice so clean. Because if we would take humans and put them in specific pathogen-free conditions and then compare them with humans, the humans would not be a good model to humans. So actually, and that has been shown several times now in the last couple of years, that if you have a mouse in a more natural environment, then their immune responses, then they look much more like humans actually than the standard mouse. So actually, I think because the people don't like variation, because if you keep them clean, the variation goes lower. So you can get nicer bars and standard deviations in the papers. We are going into models which are cleaner and cleaner and cleaner. And because of that, I think this is an important reason why they cannot be easily translated to humans. Whereas if we would keep the old dirty labs, actually they would be much better as models for humans.
Thank you so much. Thank you very much to the three of you. I think this was really a very interesting session. I think that we discuss aspects of mRNA vaccine which are not usually considered or not sufficiently considered, and as we heard, which might be very important for the future. We still have to improve those vaccines for several reasons, and I think that you offer an interesting perspective on possible avenue for these further improvements. I would also like to thank those who were following us. Others will be able to look at the recording of this webinar. We are just on time, and again, I would like to thank everyone for attending this webinar, and I wish you a nice day. Bye-bye.
Thank you very much.
Thanks, bye.
Bye.
Bye-bye. Bye-bye.